Chapter 5 is about the evolution of biodiversity. In your introduction story, they discuss the Spanish flu outbreak that occurred between 1918 and 1920 that had devastating effects on those who were diagnosed with the disease. The mortality rates estimate between 20 million and 100 million worldwide, and 600,000 people alone in the United States are suspected to have died from the disease. As scientists were trying to combat the disease, scientists in China found that the roots of a particular plant had beneficial properties to fight the flu. This plant was the Ferula asafoetida, which you can see in the top corner here. This plant had a very pleasant smell when it was cooked, but the raw sap from the roots had a very foul smell, and that's where it got the nickname dung of the devil. The virus that caused the Spanish flu was an H1N1 virus, which is very similar to the virus that caused the swine flu outbreak between 2009 and 2010. You were probably in middle school when this virus was very popular. A lot of students missed school, a lot of teachers missed school because of the swine flu. During this time, scientists in China recalled the effectiveness of dung of the devil, and they found that the plant has very strong antiviral properties, and these properties are even stronger than contemporary antiviral drugs. The dung of the devil has the potential to produce effective pharmaceutical drugs to fight the H1N1 viruses. But it's not the only organism that we have extracted life-saving drugs from. Willow trees, as we see in the second picture here, are the original source of salicylic acid. The rosy periwinkle, which is your next image moving down, which are found in the tropical forests of Madagascar, are actually the source of two different drugs. These are used to treat childhood leukemia and Hodgkin's disease. The May apple, which is the picture at the bottom, is an herb found in eastern United States, and it's also the source of two anti-cancer drugs. Not shown, we can talk about coral reefs, including sponges, corals, and sea squirts. All of these have anti-inflammatory, antiviral, and anti-tumor drugs. In fact, of the most promising current candidates for new drugs, 70% were first discovered in plants, animals, and microbes. So, what's the problem? Well, many known and suspected sources of drugs, pharmaceutical drugs, are being lost to deforestation, agriculture, and other human activities. In addition, we are losing the indigenous people who understand and have some knowledge of how these natural drugs work, because they are being forced to relocate. So why should we talk about this now? A lot of organisms on Earth are suspected to have properties that are beneficial to humans, but we have not been able to screen all of the organisms on Earth for the potential for useful drugs. So let's continue with the chapter. Earth is home to a tremendous diversity of species. When we talk about diversity of an area or of Earth, we need to look at three factors. Ecosystem diversity is the variety of ecosystems within a given region, however that region is defined. It could be defined as New Jersey, or it could be defined as an area larger like the United States. Then, getting a little bit smaller, we talk about systems diversity. This is the variety of species in a given ecosystem. So we're looking at the different organisms, the different species that are living within a given area. Moving on an even smaller level, we're going to look at one species individual, and we're going to talk about the genetic diversity. So again, this is the variety of genes within a given species. So here we're looking at the DNA of a species. So you're looking at the diversity, why these chipmunks, in our example here, do not look completely the same. All three of these scales of biodiversity contribute to the overall diversity on the planet. How many species live on Earth? Well, in order to answer this question, we need to be able to define what a species is. A species is a group of organisms that is distinct from other groups in terms of size, shape, behavior, or biogeochemical properties. They must be able to freely interbreed with other individuals and produce fertile, viable offspring. There are some scientists that challenge this definition of species because there are certain situations that have come about due to human influence. For example, a donkey and a horse bred together will produce a mule. As of the current definition of species, a mule is not its own species because it could not reproduce on its own. It is not fertile. In addition, we have things like ligers and tie-ins that are being produced in laboratory settings based on human activities. The difference between a liger and a tie-in is just whether or not the male is a lion or a tiger. These individuals often cannot reproduce on their own. However, scientists have been able to extract their sperm and egg and be able to produce offspring for the ligers and the tie-ins. But again, this is not something that would naturally happen in nature. Problems occur when we try to estimate population size. You are going to be doing a lab where we are going to be practicing the Shannon-Weiner Diversity Index to determine how diverse an area is. But when we try to identify the number of individuals within a population, we can have some difficulty. So this is often a result of organisms being too small to locate, organisms that are only active at night, and organisms that live in inaccessible locations. To date, we have identified approximately 2 million species, although the current estimates for the total number of species that are on Earth are between 5 and 100 million, which is a very large range. As you can see in this picture, these are just examples of some of the biodiversity that we have on Earth. For the most part, these are things that you can see with your naked eye, with the exception of our friend the dust mite right over here. Let's talk about some definitions that are going to be helpful for the lab that we are going to be doing later this week. There are two measures that help to determine diversity: species richness and species evenness. Species richness is the number of species in a given area, and it helps to give you an approximate sense of biodiversity as it helps to tell you the number of different species that you have. Species evenness is the measure of whether a particular ecosystem is numerically dominated by one species or are all represented by similar numbers of individuals. And that is what's being shown in the figure at the bottom here. In the community, on the left-hand side, community 1, we have high species evenness. All of these species are present in similar abundances. So all of these trees are present at about 25% of the total population. In community 2, we have low species evenness. So we have one type of tree, type A, that is dominating over the other types of trees. It's very important that we evaluate species richness and species evenness together. And by looking at these things together, we can determine some human disturbances. This is an indicator for human disturbances. We also can look at the relationships among different species, and this should be a review from last year. Phylogenies are branching patterns of evolutionary relationships, and it can be depicted in a diagram or a phylogenetic tree. The phylogenetic tree will show you the relatedness of species as determined by similarities of traits. So they look at morphology, for example, bone measurements, behavior, genetic similarities. And the more similar the traits are, the more closely related the organisms are. So if we look at the diagram at the bottom, you're going to see that lizards and their relatives are more closely related to mammals because they have a more recent branching point than lizards and relatives are to amphibians. Evolution is the mechanism underlying all biodiversity. Evolution is defined as a change in the genetic composition of a population over time, and it occurs at multiple levels. Remember this abbreviation. King Philip came over from Germany singing. This will help you remember kingdom, phylum, class, order, family, genus, species. Kingdom being your broadest category, species being your most defined category. Microevolution is evolution that occurs below the species level, so within an individual or within a species itself. Macroevolution is evolution which gives rise to new species or new genre, family, class, or phyla. And speciation is defined as the evolution of a new species. On the image that you can see on the right-hand side, this is the evolution to get the modern day horse. And you can see things have changed from their leg structure as well as their size to what we have now. We need to talk about how we create genetic diversity. So genes are defined as physical locations on chromosomes within each cell of an organism. So these help to determine the traits of an organism, and they can be passed on to the offspring. A genotype is the complete set of genes within an individual. There are two processes that create genetic diversity, which again will occur over an organism's lifetime. The first one we're going to talk about is mutations, and mutations are a random change in the genetic code. This could be caused by a variety of different things. For example, mistakes during copying, mitosis or meiosis, and environmental factors. In order for mutations or mutated traits to be passed on to future generations, those mutations must occur in the gametes or in the sex cells of the individuals. All of the pictures that we see on the right-hand side are a result of mutations. Now, some mutations can be beneficial in that it will increase an organism's chance of survival, meaning they are more likely to pass on that favorable trait to the next generation. Mutations can also be detrimental. Again, connecting back to evolution, if mutations are detrimental, the organism will often not reach sexual maturity, will often not pass those traits on to the next generation. And they can also have no effect, in which case the trait, because it is not beneficial nor detrimental, does not increase or decrease an organism's chance for survival and may or may not get passed on to the next generation. The last piece is recombination, and this is when chromosomes are duplicated during meiosis. Remember, recombination involves the pairing of homologous chromosomes, and we can have crossing over that occurs. The key here is that it does not create new genes, it only creates new combinations of genes. Please stay tuned for part 2 of the PowerPoint.